Um, our speaker is Patrick Bedford, and his title is A Regulatory Perspective on Gene Therapy Development and Commercialization. I'll get to his abstract in a minute. Patrick had his education at health sciences at Western and bioethics and law at Otago. And regulatory affairs provided him with training in his regulatory specialization in novel and emerging biotherapeutics. He's worked in government as manager, senior policy analyst, and scientific and regulatory affairs officer for Health Canada, where he managed projects including documents for preparation of clinical trial applications for use of cell therapy products in humans, information and submission requirements for subsequent entry biologics, safety of human cells, tissues, and organs for transplantation. He advised senior government officials on key issues related to biosimilars and personal medicine and companion diagnostics. He led Health Canada's international and interagency discussions with United States FDA and EMA. He was senior manager of regulatory affairs at Calgene and worked to file a drug permission for chimeric antigen T receptor, and senior manager of clinical translation and regulatory affairs in the Center of Excellence for Commercialization and Research. And he developed business plans offering regulatory and commercialization consulting services. He's the founder and principal consultant to weCANreg Consulting Group. He founded this last year. He's several publications. The one that caught my eye is his co-authorship of a title of Human Gene Editing, Revisiting Canadian Policy, that was in Nature Regenerative Medicine. Now in Canada, you can't just make up drugs in your bathtub and sell them to people. Well, actually you can. It doesn't work out very well, and we have a lot of regulations. This is part of being a real country with a real government, that things are done by law and by regulations. Regulations can include all kinds of things, including ethical issues. These days, there are plenty of ethical issues coming up regarding who's going to get vaccinated. Patrick writes, as biotechnologies continue to advance, discourse moves from can we modify human genomes to should we modify human genomes to how, when, and under what circumstances should we modify human genes. During this discussion, we will look at how Canadian regulations manage emerging novel technologies, CRISPR, which have the potential to change disease progression in people today or future generations. Okay, Patrick, it's yours.
All right, and I'll first test my microphone. Did I successfully unmute myself? I hope. Good. And next, I'm going to make sure that my screen is sharing. Can you, you should see a presentation, and that should be about it, yeah?
That's
great.
Perfect.
All right, well, thank you very much for that introduction. And I have to say, I'm excited to speak to this group. A lot of presentations that I do really focus on scientific and regulatory technical expertise and trying to train people to do regulatory. But this time, I think I listened to some coaching, and I know that you have a very broad audience with a lot of different levels of expertise in this area. So I'm trying to hit some of the most interesting topics that will lead to good discussions. I do tend to speak quickly. Feel free to interrupt me if I say something that doesn't make sense, or make a note and ask me later. I'm more than happy to delve into more detail on any of the topics. And maybe if I could just elaborate for a second on my background. I would summarize my background to say I have a very unique perspective because I have government experience. I have the academic startup support kind of experience, so early stage health product development. And I have some industry experience of later stage. So those different perspectives, not a lot of people have. So I hope that you find it interesting. And I think my presentation will really reflect the broader range of what I've kind of worked at. And if I could also just say that my new company, I'm really excited about. It's been about a year. It was born out of the pandemic. So I'll just say a quick word about what it is I do, and you'll understand the perspective that I'm coming from a little bit more, and I will touch on it again. So weCANreg is a company that's focused on supporting cell and gene therapy development in Canada. So we're really passionate about helping Canadian researchers, and predominantly in Canadian academia, who are thinking of starting up companies, so that early development. Also focus on helping some companies and researchers from outside of Canada come to Canada with their clinical trials. That's really the driver for what I do. And I really do it by doing three things. One is I help people with their plans. I orient them to the Canadian regulatory framework and what they need to do. And then I help them interact with the regulator, whether they have an issue that they can't get past, or whether it's just a kind of a pre-submission opportunity. So before you file regulatory submissions for regulators to look at your material, you can interact with them and talk about some key issues so you know what to do. So I help people with that. And then I help people accelerate their submissions. I have annotated templates and all sorts of things from my experience that have really been able to help. I can hand it to somebody and they can take it and run with it for a while and then come back and I can help with checklists and help them understand where there may be some gaps. So that's really what I've been doing for the past year. And so far it's been quite successful. I've been able to talk with over a dozen researchers in cell and gene therapy. I don't know why this is moving forward when I don't tell it to you. I hate when computers don't listen. But really, I've been able to engage with the regulator on behalf of a client multiple times and really able to move some things forward in this space. So I'll tell you a bit about that today. So part of my job is helping people understand where regulatory fits in the commercialization pathway. So I assume some of you are familiar with this. A lot of great discoveries are coming out of academia in this field and in others, and some of the things are the same, where you have whole bunch of research that's going on until you can select something that's very unique and novel. And it's a discovery. It's something that has a lot of potential. And a lot of times in academia, you work to patent it and then develop a bit of a plan. And then sometimes that involves licensing it to industry so they can develop it further. And sometimes it involves starting a company and doing more with it. And that's where academia typically operates most of the time, with public funds and other funds as well. And when things are licensed out, they sometimes get into this kind of commercialization pathway by going through industry. And that's a group that's expert at taking a technology and making it work in their facilities or in larger facilities to scale it up, to develop the product and make it a little bit better. And I'm talking about drugs at this point. And then plan for regulatory opportunities, and then file regulatory submissions so they have an approval to sell it. And then there's the marketing aspect where you plan to get reimbursement and then you have a launch and then you follow the product throughout its life cycle. So that's the typical drug development or commercialization pathway. It's not perfect. It's never linear. It's never this clear. And what I would say in cell and gene therapy in particular, where it's a novel technology and you don't know what the regulator is going to think way down that pathway, I think people are operating with what I call regulatory risk. If the regulator says, no, you can't use that type of technology in that way, or you have to do these different studies to show that it works, you risk redoing things. So what I end up doing half of my time is advocating for this. And you can see I superimpose some of my services, my consulting services, over top the commercialization pathway. And I try and get people to think about regulatory at the strategic planning stage of development. And then I get them to think about it and touch on it again when they're doing their tech transfer, whether they're doing it or whether they're having somebody else or they're licensed it out to a company. Someone's got to think about regulatory implications when you're doing tech transfer. And then when you develop your product, when you make these changes, reconsider what the implications are again. And then you really should start engaging regulatory consultants at the planning of the interaction phase. So I'm just mentioning it because I think, especially in cell and gene therapies, this is extremely important. And I'll be touching a bit on some of the activities that you do throughout this commercialization pathway as we go on. So that's just a few introductory comments. And then Milton went over my abstract. I think, as I said, I tried to stay away a bit from some of the technical topics, and I want to skip to what you might find interesting. And I look forward to exploring those further during our discussion. But it's really themed around the federal government response to emerging biotech. And I think what you'll see are things like CRISPR and other genetic editing technologies are, although very exciting and targeting new unmet diseases and things like that, in some sense, they're just another tool or technology that can fit into the drug regulatory pathway and lead to new opportunities to treat patients. But as you'll see, I think there's currently zero chance to change future generations using CRISPR or these technologies, and I'll explain why. So the drug regulatory perspective can really advance these discussions, I think, because rather than talk about whether we should, it's really exploring more details of how we can move these forward. So I'll go over at a very high level what the drug regulatory context looks like in Canada and where gene therapies fit. I'll talk about developing regulatory policy for emerging technologies, how that's done. Then I'll dive into a short history of how regulators have responded to some other emerging technologies, and then how they've responded to germline editing. And then I'll talk about gene editing, and then I'll start throwing out some ideas for what could be next. And that hopefully will lead to some really interesting discussions. So Canada's drug regulatory framework. This is a reminder probably for this group, but when I'm talking about drug regulations, I'm really talking about the federal drug regulatory activities. The federal government regulates the product, what can be sold in Canada, what claims you can make, and how they can be manufactured. Whereas in the drug world, the provincial governments are responsible for administration. That's running the hospitals and the practitioners who are delivering it. There's other ideas like off-label use and things that fall under provincial jurisdictions that I won't be touching on. So from a federal government perspective, and speaking about Health Canada, that's the drug regulator. And then there's a branch in there called the Health Products and Food Branch, which is responsible for regulating drugs, whether they're pharmaceuticals or biologics or other biotherapeutics. So things made using living things or involving administration of living things. Or medical devices. All of these are done in the Health Products and Food Branch within different directorates. And the mandate there, and the government's very mandate driven, is really two-pronged. And people generally know the first prong, which is they help Canadians maintain and improve their health. And they do that by restricting which products can get on the market. And they do that based on a benefit risk decision, which is informed by high quality science and research from the sponsor. So that's the first prong. The second prong that people forget about is the government has a mandate to help Canadians make informed, healthy choices. So that means that not only do they allow something on the market or not, but they're very interested in what the label says about it. And they monitor that throughout the lifespan, and also make sure that additional information about safety is collected and communicated to Canadians so that they can make decisions about what type of health products to take. And they do that with some tools, and I'll be using these. And if I say anything that's unfamiliar, please catch me and stop me. I don't know if we can raise hands or someone can interrupt, but really there's three main types of tools. There's the act or the legislation, there's regulations, and there's policy. And the act that I'll be talking about for the most part is the Food and Drugs Act. And these kinds of legislation are really setting out what prohibitions there are. The federal government has powers over criminal activities, and that's where this gets its authority out of the Canadian Charter says that the federal government can regulate criminal activity, and with that constitutional power, they created the Food and Drugs Act. It says what you cannot do certain things. Regulations say how you can go about demonstrating that you're not doing something illegal. And I'll show how this works with an example later. And then there's this other type of tool that's called a policy tool. That can take many forms. I'll be talking about guidance documents, but there's also policy statements and position papers and things like that that help people interpret the regulation for consistency, but they don't have the strength of law. The Act and the regulations do have the strength of law. So I mentioned I'll go through the Food and Drugs Act a little bit. I'm doing this at a very high level. This is about a 35-page document, I think, of legalese. But ultimately, from a drug regulation perspective, what the Food and Drugs Act says is, thou shalt not manufacture under unsanitary conditions or adulterate a product. Thou shalt not misrepresent the product. So you can't make false or misleading claims about something's safety or effectiveness. and and Health Canada is able to create regulations to say how someone can go about demonstrating that they're not doing that. So those are the three main things that I would pull out of the Food and Drugs Act that are relevant to our chat today, and relevant to gene therapies. And by the way, I will forget, but gene therapies are a drug. So when I say gene therapies, it's a subcategory of drugs. So when I talk about drugs, they apply to gene therapies as well. And a little bit more about the food and drug regulations. So first of all, the food and drug regulations set out a pre-market approval scheme. So that means that you can't distribute or sell a product for clinical trial purposes without first having Health Canada say, yes, you're allowed to do that. And same thing, after you've completed clinical trials, you can't sell something to the general population or allow somebody to be able to receive it in Canada without an approval from Health Canada. So that's what I mean by pre-market approval. And then when you dig down a little bit further, I don't know why this is switching, I'm sorry. When you dig down a little further, the food and drug regulations, which are a couple hundred pages, the relevant sections can really be broken down into 6 relevant sections. And I won't go through them in detail, but Division 1 really talks about the fact that drugs in Canada have to have a drug identification number on their label. So that means anytime you go to a pharmacy or you get a product from a doctor, you should be able to get this 6-digit code that says, here are the numbers, and you could go and look that drug up with the drug identification number, and you can get more information about it. Division 1 also requires people to report adverse reactions, and that applies to companies that distribute it. So they have a legal obligation when somebody makes them aware of a safety concern that they tell the regulator. And there's some other information there. Drugs are also regulated by way of the place where they're manufactured has to be registered and licensed with Health Canada. And they're licensed against standards called good manufacturing practices. So Division 1A and 2 work together to make sure that things are, as I said, under the Act, that they're manufactured under sanitary conditions that are appropriate to make a safe and consistent product. And then, there's a special section for biologics. Gene therapies are a biologic. And this is just additional requirements to have lot release tests sent to Health Canada, because there's a lot more variability with biologic drugs, and the regulator wants an ability to make sure that products are consistent. So that's another regulatory mechanism that may be of interest to you. And then the clinical trial requirements are spelled out, and that requires someone and to file a submission, a clinical trial application to Health Canada before conducting studies. And they have to issue a no objection letter before someone can move forward. And then I mentioned for all new drugs, you have to provide enough quality, safety, and effectiveness data under Division 8, or the new drugs division of the Food and Drug Regulations. Quick note about pharmaceutical products versus biologics. Pharmaceuticals are really well characterized. That means you know almost exactly everything that's in it and in what amount. And it's really tight specifications. That means there's not a lot of variability. Biologics are quite different because they're grown in living things like bacteria, for example, for monoclonal antibodies, or cells. And they're since we don't know exactly what's in every drug, we have a good idea of about how much is in every drug, but consistency in how it's processed becomes so much more important. So that's why they say the process is the product for biologic drugs. There's your crash course on the Food and Drugs Act and the regulations. And now just a quick note on policy. Don't worry, you're not intended to read this probably tiny text on the screen, but this is to remind me to talk about when we think about regulatory affairs, really it is a pile of documents. It's a pile of guidance documents and policies that you need to understand. So that's what's on the left there. And I have Canadian snapshot of the cover letter of some Canadian guidance documents and some international guidance documents. And then understanding all of those, you're required to put together a whole bunch of documents to support what it is you want to do in Canada as a drug sponsor. So this is an example of a clinical trial application content plan. These are organized in a very specific structure. So it's best visualized as a folder with a bunch of documents in it, and those folders are kind of predetermined, and that helps the regulator find everything. And that's what the common technical document, or the CTD triangle in the middle, is showing. And I say this because a new drug submission can get up to be 500,000 pages. So it's really hard to wrap your head around how that all goes together without having this visual. And that visual shows how anywhere in the world that follows ICH, which is the International Council of Harmonization, will have the same submission that's purple and down. So only the tip of that triangle is regional specific. That's the intent anyways. And the rest is structures. So you have manufacturing data in one place, and the purple documents are summaries. The blue, green, and other blue on the bottom are for your manufacturing data, the actual results, the preclinical safety data, and then the effectiveness or efficacy data on the bottom right. So if that helps, I hope it does, understand a bit more of what regulatory affairs is. It's putting these together and understanding how they're all interrelated. So there's your overview of regulatory. Regulatory in what, 15 minutes. I'd be happy to go into details about them, but just so that we make sure we move along, I'll get into how those policies are actually developed now. But first, a quick story. So as you know, I have an undergraduate degree in health sciences and a master's in bioethics and health law. And right after I graduated, I got a job at Health Canada with the regulators. And I think my first activity, they thought it would be very interesting if I gave a regulatory perspective, or sorry, an ethics perspective of regulatory decision making. And I remember I put a lot of time into it, and I described things like the principles of bioethics as described by Beauchamp and Childress, which was one of the textbooks that I had in my undergrad, and applied it to regulatory decision making. And I made the point that regulators say they make science-based decisions, but ultimately benefit risk is going to be a normative judgment. You're going to say whether something's, well, just subjectively good enough or not, and you have to put on your hat of a patient or a doctor who wants to administer it and think, is this the right thing? Should we allow people to do it? And that presentation did not go over well at all. Someone put up their hand and said, well, why don't we just let the Catholic decision decide what drugs are approved in Canada? And that was a really difficult time because recent graduate, I thought I did a great job, but I think ultimately I learned a lot about policy development there, and I debriefed with my supervisor to say, well, what really happened there? And he I think the line that I remember most is he said, well, policy is ethics, but with a useful outcome. And I was insulted at first, but then I kind of understood, because I think I was ready to raise issues and say that we need to do a bit more, but I wasn't offering any solutions. And policy is inherently solutions oriented. And I ended up doing more reading, and I found the Cabinet Directive on Regulating, which is available online if you ever wanted to look at it. And it has principles not unlike principles of bioethics, but a lot of them were saying how you go about finding solutions and proposing them and documenting decision making for government. So there's kind of my experience of moving from ethics to policy. And there is an element that I believe that policy is a really useful endeavor. It should take into account science and everything else, but also factor in some ethics and some practicality and some public health decision making and things like that. So, and of course, there is another tool, and it's the process for making policy in government. And Health Canada also publishes this decision-making framework. And don't worry if you can't read the text. Obviously, if you just found decision-making framework, that's the title on the left. And on the right, you see a summary of the decision-making process. And decisions are made by first identifying the right issue. This is actually the most difficult part in policy development, if you ask me, and the most important part. And then assessing and the risks and benefits of the current situation, then identifying potential other options and the pros and cons for each, then selecting a strategy to move forward, and it should be the least burdensome approach is one of the principles that's also spelled out in there, and then implementing the strategy, and then monitoring and and evaluating its effectiveness. All along, the government's expected to involve interested and affected stakeholders, and I think they're pretty good at doing that. And anytime that I was part of developing a policy, that was some of my favorite parts, was reaching out and saying, look, we've identified this issue. We think these are the potential problems, the implications. Here's some options, here's the pros and cons, and doing kind of those presentations and brainstorming sessions and working it through was really very interesting. So that's how the process by which policy is made. And I would say that the key challenges for the regulator in regulating emerging technologies is maintaining credibility and trust by keeping pace with these emerging technologies. If they cannot allow something to move forward in an appropriate way, they get a lot of feedback. And I think they lose some credibility and trust because patients are waiting for drugs and new technologies to make it to their clinics so that they can get better. So that's a key challenge for the regulator. It's really difficult because you never know exactly what the future holds. Another challenge is balancing the known benefits and risks in the context of uncertainty. And remember, the regulator is well aware that there's benefits and risks of a drug. There's always risks. Otherwise, it's doing nothing, most likely. And there's always the benefits. But then you have to weigh the benefits and risks of a product against the benefits and risks of not having that product. So that gets a little bit more complicated. And then you have to do it in the context of there's other treatments out there with their own benefits and risks. So it's actually quite a juggling act. And I'd say the regulator also really struggles with becoming a barrier or an enabler. So believe it or not, regulators do realize that they're very important in Canada's economy. If there's undue regulatory burdens, new drugs won't come to Canada. But if unsafe drugs come here, then people won't trust the drugs that are coming. So they really have to be really careful with this. And I would say, and I'll say more about what I think enabling regulations actually allow them to do and what they look like in the next slide. So I would say, and this is from a presentation that I gave, I think in 2015 or 16 when I was still at Health Canada. Health Canada thinks what they have and what they should develop are enabling regulations. So they're defined as they're performance-based. So they're non-prescriptive. They say some standard must be met, and they'll, for example, but they won't say how you have to meet it. And that's really important. In the past, for things like the measles vaccine, they would say, well, you have to scrape a horse's scabs in this way and have it at this temperature. And it was too prescriptive and it didn't evolve over time. So what they do now is they say, you have to make sure that you manufacture under appropriate circumstances and you have evidence to support what those appropriate circumstances are. And then it's up to the sponsor to figure out how to do that and then follow their own rules that are now enforced by the regulator. So regulations have to be performance-based and they have to be flexible. So what applies to pharmaceuticals didn't apply directly very well to biologics. So a different division had to be written. And if you had to write a new division for every type of new technology, when you had nanotechnology, when you had xenotransplantation, when you had, it would become burdensome just to have so many different regulations. So the goal is to have something that's flexible and applies to a wide range of technologies that are foreseeable. And finally, enabling regulations have to be clear and supported by policy. And that just means so that you can actually predict what the regulator will expect. So you can't just give someone a blank check and say, you have to fill this out and it's up to you to make sure that your bank will be able to clear the check. They actually say, well, and the service is about this much money or whatever. I mean, that's a money example, but the idea is just having open, transparent regulations isn't enough. People actually want to know what the regulator expects. So there's your crash course or the top high level, I guess, of how policy is developed from my perspective and from my time working at Health Canada and now trying to influence policy from outside of the government. And so now I'll talk a little bit about the history, or I think over the past 100 years, I'll only hit the top level, don't worry, to see how regulations have actually evolved over time. And again, this is tiny text. I don't expect to read it, and I'm sorry, it's just more of a visual for me, and hopefully interesting to you. And if you want to look at it later, it's there. But what I would say and focus on is the first half of the century from, 1875. This is the Food and Drugs Act and how it's and regulations under it that have come out over time. Just a snapshot of some key things. And I'd say, the first half of the century was really marked by reactionary measures, from alcohol adulteration to teratogenicity of thalidomide. So a drug that obviously some of you may know led to malformations in utero for women who were taking it and became pregnant. And all the regulations based on negative experiences were reactionary because they would make amendments based on lessons learned in tragedies most of the time. But they did improve and hopefully prevent things from happening in the future. And I'd say it wasn't until, I guess, maybe the 1980s when lessons learned and working, we switched from being a bit reactionary to more precautionary measures. And you'll remember in the 80s is when a drug, blood, was tainted by an emerging virus that people didn't know a lot about yet, HIV and also hepatitis, but and the regulator didn't act quickly enough. So what they did at that time is adopt a precautionary approach to regulations. And the precautionary principle says that regulations should not wait for scientific certainty before you act on health risks or potential health risks. So there was a fundamental shift in regulation there that I think is still with us today, and particularly in biologics, which is relevant to gene therapies. It's been internalized quite a bit at Health Canada. And rightly or wrongly, I think this approach is why Canadians can't move forward with things like xenotransplantation or germline editing. It's the fear of the unknown that could lead to a negative risk, and Health Canada is willing to act on that fear to prohibit the potential risk from emerging. And that precautionary approach still exists. But I think there's also a bit of a flavor of a risk-based approach now where you can identify a potential risk and accept that it's there, but still figure out how to move forward if the risk is low enough, the theoretical risk. That's why I say we also have a risk-based approach. And I'd say things like our transplantation regulations, medical device regulations, which actually classify risks from 1 to 3, low to high, and have different regulatory burdens or requirements associated with those, is emerging. The natural health product regulations are a risk-based new framework. So I'd say we've moved from being reactionary with developing regulations to being a little precautionary, and now we're at a risk-based, and I'll be asking maybe what's next later. So here you have a bit of a visual where I charted the kind of amount of regulatory burden over time, and it was increasing as we knew more, and we knew that the federal government had to prevent certain risks or people from benefiting from making false claims. So all of those activities led to higher and higher regulatory burden, but it didn't happen in a linear way. It happened, and I have about in the halfway through the century, we have those examples that made it necessary for people to file drug submissions before they get approved and to address safety and to do more clinical studies. So the regulatory burden was going higher. And then we had the 1980s where there was the Creever Commission against what happened with tainted blood and things like that, until ultimately I think we got to a peak of regulatory burden with xenotransplantation and germline editing. And as you'll see, I think both of those are the peak because you cannot get more regulation than that. I'll use an analogy later that the regulator came out with a sledgehammer from their tool shed and just said, you cannot do this in any shape, way, or form because of the potential risks. And I think we might be backing off from that a bit more into a more practical and sustainable way of managing regulatory burden and responding to emerging health threats. And sorry about the question mark there, and you may wonder what's an advanced therapeutic product or what are these regulatory sandboxes? And that's why I say I think we hit the peak in maybe in 2002 to 2008. Last year, Health Canada had a new legislative amendment that allows them to kind of build the plane as they're flying it. They now have the authority to write product-specific regulations and exempt certain products from any and all part of the Food and Drugs Act and regulations if they're so novel that those regulations shouldn't apply. So I'll get into that. The other thing that the regulator is doing, and they published a report not long ago, is they're working with other regulators now and showing some international leadership with groups like ICMRA, which are the head of the regulatory departments in some partner countries. And they're conducting regulatory foresight to see where technologies are going so that they can prepare regulations before they're actually needed. And they recently published this report. It's called What We Heard in Drug Regulation, to say what they think is emerging. And there's, sorry. There's 6 technologies that they highlighted there. I haven't, the next slide makes it legible, but I encourage you to look at this to see what's next in drug development in the world according to the regulators. And what they say are these technologies we'll likely see in the coming years. So we'll see the use of big data, so that, and artificial intelligence, they probably go together quite a bit. We'll see 3D printing, probably at the bedside and in hospital spaces and in novel ways, which will decentralize manufacturing from these big facilities that are being made by device and drug developers right now. We'll see new ways of delivering drugs. So these are maybe nanoparticles, or they're going to be like just novel delivery devices, new combinations of drugs and devices. And then we'll see advanced cell therapies and gene therapies, and these are linked, and I'll talk about them more. And that's more in my world, and I was really interested to see what they think is coming up. And now, what I think remains to be seen. I tried my hand as an artist. Now that we're at home and working, many of us are working from home. I thought I'd take an extra minute with my kids to draw out some things and have a little bit of fun. But the point here is, I think Health Canada and any regulator, their natural reaction when they have an emerging technology like gene therapy is they reach into their toolbox and they see what's there. And in this case, they have a hammer, the Food and Drugs Act and regulations. And when you have a hammer, of course, everything you see is nails. And cell and gene therapies are considered drugs, so they're nails, and they start hammering away. Of course, cell and gene therapies do have some unique aspects to them, and I think they might look a little more like screws. And at some point, you have to ask whether the regulator is going to say, you know what, I need the proper tool for the job, and then they might have to make a regulatory amendment. So there, I guess I've gone through, I guess, how our general context for regulation, and then how regulations and policies are developed, and then what it's looked like a little bit over the past. So now I'll jump into what I guess was promised, is talking a bit about CRISPR, or at least gene editing. And I'll tell you right now, I split up this gene editing section into 2 parts. The first part is going to talk a bit about germline gene editing technologies, and the second part is going to talk about gene therapy technologies for reasons that will become obvious. So regarding germline editing in Canada, can't do it. It's illegal. The regulation, I think in 2004, just said that no person shall knowingly alter the genome of a cell of a human being or in vitro embryo such that the alteration is capable of being transmitted to descendants. And remember, we have the Food and Drugs Act and regulations. So theoretically, if you're doing it for a therapeutic purpose, it would fit under there, except this overarching requirement for reproductive purposes, which sits under the Assisted Human Reproduction Act, another group that was in Health Canada as well, said, regardless of what you're doing, if it's capable of being passed down to descendants, then it's illegal. You cannot do it. It's prohibited. And there's no regulations to say how it's legal. And I think it's a touch ambiguous. I mean, I think we know what the intent was. It was about the right of the embryo, and it had a lot of ethical connotations to this. I don't want to go down that rabbit hole too much. It's fascinating, and I think it's worthy of discussion, but I don't claim any expertise there. But what I think happened is they missed an opportunity to clarify, well, what if the intent is never to implant and grow the embryo? It's no longer necessarily capable of becoming a human, but theoretically it is. And that's actually how they've intended it. So they prevented any research on embryos or semen or ova or any germline piece from being edited, even in research. So naturally, I think there were some responses to this. And this is, I think, what Milton mentioned. I have a paper. I'm an author on 3 of the papers here, the last 3 bullets. But there was a lot of pushback then, a lot of discussion, and there's been a lot more recently because other regulatory jurisdictions aren't doing the same thing as Canada. So I'll go over this quickly. There's at least 4 publications involving top tier Canadian researchers. These are tier 1 ones like Timothy Caulfield and Bartha Knoppers in the area of ethics and health law who take issue with the prohibition and think that it should change moving forward. Of primary concern is the tool selection. So the government really used the biggest sledgehammer in the shed, as I said before, a blanket criminal ban for prohibiting this, when other governments use more refined tools to say what's allowed and how and other things like that, and really tried to get to the nitty gritty of what's wrong and what's right and what should happen. Of equal concern, I'd say, is that the impact of legislation is that leading Canadian researchers just go elsewhere to obtain data and conduct research that could inform future regulations in this area, which may be more appropriate. And ultimately, a more nuanced concern is that gene therapies, which is advancing, as I will discuss next, may actually inadvertently trigger some of the prohibitions here. So someone who's doing research that could be approved through a clinical trial by one arm of the government might actually inadvertently change the germline. I think it's a theoretical risk at this point, but when you think about regulations, there's no consideration for how these interact. So I think there's a lot of problems with the ban, even if somebody were to agree from an ethics perspective that it's the right ban. I think there's some additional work and clarification that can be done by government in the space. So I'll jump into more of my wheelhouse here with drug development, and that's what can move forward, and I'll talk a bit about gene therapies. I should start the section by stating that gene therapies are drugs. I'm just going to remind you, according to federal regulations, but there are no regulatory or policy definitions in Canada for what a gene therapy is. There's no special frameworks, there's no special pathways, there's not even a guidance document on gene therapy in Canada. So what you're going to hear from me is from my experience in supporting people who are developing these, and from what I read in federal regulatory requirements in the US or international, but nothing in Canada. So a quick terminology overview. I don't think I'll use acronyms or anything, but still, these might be really interesting, and I'll just touch on them, and you can pick up if they're interesting, and we'll discuss later. But there's tons of ways to subcategorize different gene therapies. You recently heard about CRISPR technology in the media, and may even read about viral vectors and how they're used. And you'll pick up on terminology like indels or editing or additions of genes or deleting genes and things like that. I'm not going to go into detail about these, but first I'll just say that there's two strategies for editing genes in cells. First is you can take a cell out of the body, and then you can edit it with some technologies so that it can do what you want through engineering the cell, and then you can grow it and do whatever, and then inject it back in for a therapeutic purpose. That's a type of cell-based gene therapy. Or you can take something like a viral vector to introduce genes into the body that will have to go through the body, find a target, make the edit, and then get the desired result. So there are two different strategies. And it's important to understand that we have to talk about them separately a little bit, because there's separate risks associated and benefits of different technologies. And then there's also three main technologies that have been around. Two of them have been around for not a short period of time. So there's zinc finger nuclease, there's TALENs. These are technologies that, like CRISPR, have a way of targeting genes, a way of editing the genes, and they're all based on kind of an enzymatic unzipping of the genetic code and then modifying it. And there's multiple strategies for curing genetic diseases, which each depend on the disease that you're talking about. And sometimes this is easiest and talked about like a kind of find and replace, or a cut and paste, or just a cut and then let natural, natural activities repair the genes. But so anyways, lots going on in this space, and it can be subdivided in a lot of different ways. But a theme for all of them is some of the safety concerns. So first of all, for anything that's administered into the body using a viral vector, or even for things that are cells that are modified using a viral vector ex vivo outside of the body, there's the concern of viral shedding. So viruses are really good at taking their DNA and injecting it all over the place. And just because it goes into a person doesn't mean that it doesn't come back out and then potentially affect people around them. So that's a unique feature of gene therapies, where someone can, you don't just look at this from the patient's benefit risk, you look at other risks around them, the people around them, or public health risks. Another gene therapy unique concern is you can have a lot of unintended effects, and this can be both on target and off target. So this means if you insert something that's strips the genome open and it recombines in a new way, well, you could have an unintended effect that actually works, but have a different result. You can also have off-target effects. So you can target it all you want, but if there's something similar in the body, it can go and it can have an effect there. And the biggest challenge with these things, I think, is you get that in early first in human, but you address it, but you still don't know for certain because these all have a long-term safety implications. These can be adopted and kept around in a person for a very long time. And if you're messing with somebody's genes, then you could have genotoxicity or tumorigenicity, and both of these could lead to things like cancerous growth, which you have to worry about. And so there are regulatory activities like long-term follow-up of 10 to 15 years for anyone who receives gene therapies. There's also a few unique regulatory issues. And the first one's kind of an abstract one, but normally with small molecule pharmaceuticals, for example, you have a decent profile a safety profile that you understand, and you're trying an incremental benefit. That's actually one of the criticisms of industry research for a long time now, is they're looking at incremental benefits and short-term gains. But gene therapies, a lot of them are targeting rare diseases and looking to cure them. It's not with a one-time therapy. So the benefit is so significant, so potentially high, but that kind of change for a lifetime is also coming with a really high risk. So it's really a extreme benefit risk decision for the regulator. And then later on for the reimbursement body, because these are not cheap therapies. And I'll give some examples of the prices so far. Another thing is there's not a lot of good models of disease for these rare genetic diseases. So you don't get really good safety and potential proof of concept efficacy data in animals. So you're going a little more blind. These aren't established yet. So that's a challenge for regulators and people trying to navigate regulatory pathways. And then also the cost and privacy implications of following people for 10 to 15 years is a bit of a regulatory issue as well. As I said, Canada doesn't have policies in gene therapies. So all we have are sort of relevant policies, and one of them is the cell therapy clinical trial guidance. Now, I led the development of that guidance document, led the working group for it, and held the pen. And it intentionally says this doesn't apply to gene therapies, but I'll say it's sort of relevant, and it's actually one of the most helpful ones because it does tip the regulator's hand as to how they're thinking about cell and gene therapies. And as I mentioned before, there's that overlap. You can have a gene-modified cell therapy, and this guidance would theoretically apply, but the gene therapy that you inject into a person to change their gene, it doesn't apply to that. But so we have just sort of relevant policies. And in the sort of relevant policies, it highlights and says it subcategorizes potential issues and lets you think about how to address emerging gene therapies and how to plan for the manufacturing and the preclinical studies. And that's what you have to do. When you do regulatory affairs, you have to tear your brain in two and think manufacturing and clinical. And then clinical is broken down into safety and effectiveness. So the cell therapy guidance says that the regulator is willing to take a risk-based approach to the materials. A lot of the materials are extremely rare. Things like the lentiviral vectors and things like that are not able to be manufactured in the most typical manufacturing plants. And some contract manufacturing organizations may not want them in their facilities because of the potential to contaminate other products. And not only that, it's not hard to make viruses, but it is hard to purify it and to know exactly what's there, and you end up losing a lot of the products. So the cost of developing the materials required to make gene therapies is quite high, and there are a lot of unique considerations for them, and a lot of them are animal-derived as well, which introduces additional risks. Also, as I mentioned, the cell therapy guidance does say the typical drug development may not apply. Normally, drugs go through phase one studies in a small group of healthy people. Then they go to phase 2, where there's a larger group of disease population, and you get some safety data before you go into phase 3 confirmatory safety and efficacy data. But cell therapies, because they're targeting rare diseases and they're quite expensive and other factors, Health Canada has actually said, look, we probably have a early phase trial and a late phase trial. And I'll tell you, some of the early cell therapies were approved based on 10 to 20 early phase trial participants and 80 to 100 large phase or large later stage clinical trials. So not a lot of patient experience. So that's what the clinical trial guidance talks about and says, offers some guidance for. Now my quick list of approved cell and gene therapies is not too long. I've categorized them based on some of the things we talked about. So the first one approved, I think was about in 2008 or 10, was Prochymal. It was a mesenchymal stromal cell that was engineered. So this is a cell that comes out of, I think it was bone marrow. It also exists in adipose tissue fat and other things. It was taken out, it was modified and re-injected, and it was intended to treat graft versus host disease because these cells are known to and have been augmented to modify the immune system and inflammatory response. And I just took a note there, sorry about the small text, but that drug I think was reported to cost up to $200,000 a year. Hasn't been sold in Canada yet. They don't have an establishment license to my knowledge, but they did get approved based on their clinical trials. And then there's the CAR T immunotherapies. Now, when I worked at Celgene, I was involved in preparing submissions for a CAR T. So I know a little bit more about them if you have some questions, just generalizable knowledge that I can share. And these are when you take out blood cells and specifically some of the immune cells, the T cells in a person, you can put them into essentially a machine and in a controlled system. And then you can introduce genetic material to it to cause antigen receptors, chimeric antigen receptors, so genes that will help grow little surface things on the cells to help them target cancer, and actually all B cells. And that's used to treat hematological cancers. And just to give you an idea of the cost, those are reported to cost $373,000 US dollars for the one and $475,000 for the other, and that's per patient. So these are really expensive therapies, partly because they cost a lot more to make, but also obviously there's a lot of other factors when you consider cost. But obviously the benefits, what industry really wants people to focus on, and there is some really exciting results that are published about those products. But I'd say those are the gene modified cell-based therapies that are modified ex vivo. And we do have some early examples of 2 gene therapies that are actually approved in Canada and able to be injected, Luxturna and Zolgensma. These are, they edit and replace a gene using a working copy, using adeno-associated virus that's injected into the person carrying this viral load to modify genes. Nothing using CRISPR has been approved yet, by the way, anywhere in the world. They're at the investigational stage of development at this point. And the gene editing actually has an even wider range cost of $425,000 per patient, up to $2.8 million per patient. So very exciting things happening in the space, but obviously cost is going to be a huge issue, and we could probably chat about that. That'd be an interesting topic as well. So I guess that brings us to what's next. As I mentioned, I think that we're going to see very exciting cell gene editing technologies that are changing not the genome, but that are introducing genes into patients, typically with rare genetic diseases. And the pathway that we can expect can get more efficient and maybe lead to more cost-effective treatment alternatives. And they'll go through discovery. Most of them are at that stage still, but they'll be targeting unmet needs. So these are orphan drugs. There's a special pathway in the US and in the EU for this type of product. Health Canada is considering what to do and how to help orphan drug targets a bit more. We'll see these products get developed further. They'll go through their early phase trials, their later phase trials. They'll then go for market approvals. And there is a pathway that a lot of these will go through, which is the priority review, which just means a shorter time period rather than 300 days for Health Canada to review the final culmination of all the data. They'll have 180 days, so 6 months earlier, or the notice of compliance with conditions, which is a special pathway that accepts that some drugs, because of their exceptional need, should be approved just based on complete safety data, but only promising efficacy data. And typically there's a biomarker that people can rely on to say, well, approve us now and we'll give you more data later. It's almost a promissory note. And then we'll see people following these up for 10 to 15 years and probably reevaluating them along the way. And there's new authorities to do that. So that's already happening. And germline editing, as I mentioned, in Canada, is going to not even get past discovery. If somebody finds out that they have an ability to modify the human genome, then they're going to have to go to the States or somewhere else where there's some ways to move some of that research forward. And some trends I see, just in case there are discussion points that you want to touch on. I think we're getting increasingly accessible technologies. That's the most exciting thing that I understand about CRISPR, is rather than, it's relatively easy compared to other gene editing technologies to do, and relatively cheap as well. So this is the type of thing people say, well, you can actually even do it in your garage. I mean, you'd never be allowed to if you're ever going to introduce it to a person, but it's relatively simple to put together the construct. Also, right now, I think things are relatively crude in their application, and while they're early in development, so there's a lot of flexibility before you get to clinical trials, and then where they're allowing some experimentation, because there is no well-trodden path. The regulators are quite receptive to novel ways of doing things. I think that'll tighten up a little bit over time. The other thing I think right now is there's the clinic is leading the manufacturing. So normally what happens, you develop a drug and you develop a really good one, and then you do a little more studies, you make it a bit better, and then ultimately you lead up to a clinical trial. Whereas because these are targeting unmet needs, and there's such a huge unmet need in some of the disease populations, that as soon as you find something that could work and you've done the bare minimum of what you have to make sure that your product is what you expect it to be and it was manufactured under appropriate circumstances, then they're allowing people to start doing their studies. So that's what I mean by the clinical is leading the science and the manufacturing right now. We'll see faster innovation, and I think we've seen a lot of delayed investor interest where things are staying in academia longer and getting into clinical trials before somebody will invest in it. And that's because it's so expensive and there is such a high benefit risk that I think they've been waiting. I think we'll see that a little bit happen a little bit faster as investors get a little more educated and prepared to, or sophisticated in how they're selecting these. And I think we'll see some more precise targeting, and I hope and I think we'll see some novel regulatory approaches when things can be done to bedside, for example. So I think that's what I had for my planned comments, and I'm happy to maybe I'll turn it over to you to decide whether we're going to have a break first or if we want to just jump into some questions.
I think we'll thank you very much, Patrick. Really interesting stuff, and I'm sure there'll be lots of questions. But let's take a break of 5 minutes first, and then we'll come back with a question period.
Perfect.
Thanks.